Hi everyone, it's me, Julia Hull of Blazy Biology Productions, and I am here at our Pulliam Common Garden where we've planted some cottonwoods. Before we began the restoration project, the landowners were mining sand, which did two things. First, it created this pond that we use to water the cottonwoods and willows that we've planted here. Second, and more importantly, it prevented the highly invasive tamarisk from establishing, even though it was pretty much everywhere else. This is really valuable from a scientific standpoint because it allows us to have Tamarisk legacy soil treatment group next to a non-Tamarisk legacy soil control group.
I'm collecting non-Tamarisk legacy soil for the experiment we're setting up at the high school.
So here are some of the defoliated tamarisks. So these big ass tamarisks were here. The biological control agent and the tamarisk leaf beetle came through, defoliated them. But here you can see some tamarisk resprouts. All this dry leaves have been defoliated by the tamarisk leaf beetle. But here on this other shoot, you can see that actually they just send out new leaves after the beetles come through and defoliate them, so they're able to continue to live. Tamarisk alters some of the chemical and biological properties of the soil. We know that tamarisk can increase the saltiness of the soil, and that's what these white patches are, salt deposits. Tamarisk also decrease the number and types of root-associated beneficial fungi that native plants like cottonwoods and willows depend on.
All right, so now I'm collecting Tamarisk legacy soil.
When tamarisk is removed, or if it's killed by the tamarisk leaf beetle, those legacy effects remain for years. And that's why we're using this tamarisk legacy soil to set up our experiment at the high school.